The next presenter is Aderans Research Institute, and presenting for them is their CEO, Ken Washenik. They're located in Atlanta, Georgia. Yeah.
Thanks. Well, we are definitely something completely different than most of the things you'll hear about today and tomorrow. And maybe it's good to follow up on a fashion quote that started us on the last one, because we are a biotech group, about 60 team members in size in Marietta, Georgia, just north of Atlanta. Opened our doors in 2002, and we've been working in the area of autologous cellular hair follicle regeneration. So specifically, we see ourselves as looking to fill a part of aesthetic regenerative medicine that's very, very popular now, and that is a minimally invasive or less invasive procedural entity, in our case, for regenerating hair follicles in patients who've lost hair. So if you think of the model of wrinkles and where Botox fits between the invasive facelift and the prescription medications, so you take for hair, you've got Rogaine, Propecia. On one end of the spectrum, the other end, you've got hair transplantation surgery. So our product that we've been developing over the past 10 years, we affectionately call Ji Gami. We're a subsidiary of a Japanese company. In Japanese, Ji Gami means your hair. So we've been generating a cell product to go into the market for aesthetic medicine. So it's very different than some of the challenges you heard this morning about what do you do about reimbursement, because our reimbursement comes from our patients, right? There will be no third-party reimbursement that we foresee in this area. But the market is a large one. If you look at hair regeneration in the entire global market, it's over $2 billion. The bulk of that $2 billion, $2.5 billion, is in surgical hair restoration. So the area we're working in currently to do a hair transplantation, people take around 20 to 30 square centimeters from the back of your scalp to produce hair follicles that can be moved to other areas of your scalp where you need more hair. In our case, we take 1 square centimeter, so it's much less invasive. And yet we know, even given the invasive nature, if you look at data from the International Society of Hair Restoration Surgery, over the years, hair transplantation surgery is more and more popular with patients. So we're certain, and I'll show you at the end some focus group work, that when we get there, the product will be in great demand. So when we opened the doors in 2002, we started working on some assays to determine if cells are trichogenic. And specifically, we wanted to work in a cell culture system that was serum-free. We think that's important from a product standpoint, both from a regulatory as well as a patient customer acceptance standpoint. So we came up with something where we take this specimen of tissue, we disperse the cells into single cell layer culture, and then we culture separately an epidermal and a dermal component. We found that very important to our approach to separate these two principal components. But both components are a mixed cell suspension, not a purified stem cell population at all. And we developed what's called the hair patch assay. It's used around the world now to look at the trichogenic potential of cells, dermal and epidermal cells, their ability to form hair follicles in vitro. And so when we put these cells together, we form structures like this. These are the roots of the hair follicle around the outside, growing into a luminal space in the middle, if you will, a cystic structure. And what we find is that when we put our cultured cells together, if they're trichogenic, they undergo a process of homotypic aggregation, where the dermal cells condense and aggregate, and then they influence the epithelial cells to form a new structure and differentiate into a growing hair follicle. And all of this organogenesis occurs between 1 and 2 weeks. These structures usually form in about 8 days after you put this clear drop of suspended cells into the mouse. Now, the problem with that assay was it was not very translatable to the human condition. And early on, we spent a lot of time looking at developing architecture, scaffolding that we can put our cells in to try to orient the growth of hair follicles, because despite all of the success that we were so proud of, this is of limited utility in an aesthetic environment. So we developed a human xenograft assay, which is much more similar and enabled us to leap from the lab into the clinics. Here we took usually male tissue, men who are undergoing hair transplantation and can donate a piece of their tissue. We cultured these cells and injected it into facial skin from women who had undergone facelifts and donated a piece of their preauricular hairless skin. We grafted that skin onto the back of nude mice and were able to show that the male cells were able to produce this visible terminal hair growth on these pieces of skin. And our mechanism of action that we've been able to deduce, in fact, does not form brand new hair follicles as it did in that hair patch assay in our first in vitro assay, but rather when we inject this mixture of dermal and epidermal cells that have been cultured to optimize their hair forming potential, we find that the cells intercalate into existing hair follicle structures and reprogram them or turn them back on for hair growth. And so this system will only work in people who have atrophic or miniaturized hair follicles, like in male pattern and female pattern alopecia or hair loss or androgenetic alopecia. And the way we were able to pin down that mechanism is by injecting these male cells into that skin model and then following where the Y chromosome located. And when we do overlay immunofluorescence, we can see that we are forming chimeric hairs. So we're not creating brand new hairs, but rather forming chimeric hairs. With that concept established, we went to the FDA in 2005, ran our phase 1 in 2006 through 2008, and then entered a phase 2 stage of development, which is where we are today. So we're in the latter stages. We've enrolled around 400 subjects in an iterative series of protocols, looking at the ability of these Ji Gami cells to induce hair growth in men and women with androgenetic alopecia. The study design was simple. We put tattoos on the back of the balding vertex area of the scalp, injected our cells into these treatment areas, tracked those with macrophotography, left an uninjected area as a control on the back of the scalp. And we did that in phase 1 and our first few phase 2s. Later in the phase 2 development system, we started injecting this whole balding area after we would inject the 4 square centimeters that we were going to track with macrophotography. We developed 2 proprietary injection devices that will allow us to give micro volumes, 4 microliter volumes of cells without damaging the cells in a consistent, reproducible way. We started with this device, a micrometer-assisted delivery system, and have now transitioned to the Aderans Research pen that we developed so that the physician or investigator just has to depress the button and 4 microliters are dispensed into the dermis. These are intradermal injections, and injecting the cells in a way that doesn't damage them consistently in a small enough bore that the cells stay where we want them to go was a great challenge. But we think now from a commercialization standpoint, this type of device will be much more appreciated by the physicians on the other end. So this is the way our data looks. The data in this field is tracked usually by total hair count within a certain area. In this case, we chose 1 square centimeter. You need to have tattoos so you know you're in the same spot every time you take the macro photograph. And these circles highlight some of the areas where you can see on your left there was no hair, and on your right, hair follicles are now visible for counting. And that was at 12 weeks. This is an example of a subject that was injected here at the beginning of the trial, and then 1 year later, you can see the increase in hair follicles that are obviously countable. This is, if you think of the balding circle, this is the edge of the balding circle, so there are more hair follicles here, whereas here there is more toward the center of the balding circle, fewer hair follicles, and you can see the ability of these cells to turn on hair follicles that had been miniaturized and had, over time, reduced over and over the length and duration of their anagen cycle. The way we track our data in the phase 2 trial is we report our data as the percentage of subjects who grow hair. So we bring in balding men and women, we inject them with cells, we track them over time, we inject them here at the beginning, and then with no further treatments, we follow them out for a year. And what we've been showing is around 60% of the subjects we inject end up with more hair than they started with. When we drill down and look at how much hair, usually it's around 40% generate 13 or more hairs per square centimeter, which is the kind of number you see with a approved products such as finasteride or Propecia, or in South Korea, dutasteride, where they showed similar numbers to get their approval. These are the kinds of changes we see in our subjects. You can see the tattoos here in the vertex area and the increase in density in the center of the balding area. But most impressive is the size of the balding circle starts to come in, so it shrinks in from the sides. And we know that in drugs, in use of androgen prolongation agents or androgen modulators, like minoxidil or like finasteride, that's what you usually see, is that transition zone starts to shrink in. Those hair follicles have been miniaturized for the shortest period of time, and they're most receptive to any type of treatment modality. And so for that reason, in drug development, most of the studies study this dynamic area of hair loss at the edge of the balding circle. This is another phase 2 trial that we've done. We're in our 11th phase 2 trial. When I said we have 400 subjects, it's an iterative series of protocols that have 30 or 40 subjects where we try slight modifications of our cells. And again, we see here where we count terminal hairs, not total hairs, we're still coming in at about a 60% efficacy for our ability to reach hair growth in our subjects. So one of the things that we're working on now is to try to establish a set of markers. We've got a gene array analysis system where we've identified a proprietary group of markers that can help us identify whether our cells will be inactive or active, and we're hoping to continue in this phase 2 program to reach a predictive state where if we cannot get above the level of efficacy we see now, we will be able to be more predictive from an incoming tissue specimen standpoint to see whether or not a subject will be responsive. We've built a strong intellectual property portfolio to protect ourselves in this space over the years. And the process of Ji Gami, this cell-based process, is a patient would come in, the doctor would excise a square centimeter biopsy from the occipital part of their scalp. The tissue would be processed in a central facility, so not proprietary in-office type incubators, but in the central facility for about 2 weeks. Then they would be shipped back to the physician's office for intradermal injection into the area of balding or thinning. As I said, we've done a number of focus groups with both of our potential customers, the patients and the physicians. And autologous therapy is very popular with patients. The fact that this would work in both men and women is a big advance. Much of what's available now works specifically for men. And in this case, we're looking about, from age 50 onwards, about half of the population of the U.S., so 60 to 70 million men and women, so around 30 million women. And right now, they're really underserved. In hair transplantation, at least in my practice, 90-some% of the patients are male, and only around 6% of the procedural patients are female. So something that will work in women is very attractive. It's also very attractive to physicians because it's an office-based procedure. They have control, they don't have to worry about compliance, and they're able to treat the patient, know that the treatment was put in, and then follow the progress. Our studies have only tracked the hair growth for 1 year, and only with injections at the beginning of the trial. The possibility of sequential treatments down the road do exist also. So that's it. So that's my daughter with her own hair problem. If you have any questions, I'm happy to answer them now. Otherwise, myself and Vern Liebman, our COO, will be here all throughout the day. So thanks very much.